It's a pleasure for me to introduce the next session, and the speaker for the next, the moderator for the next session is Professor Alison Marsden. Professor Marsden has been at Stanford for several years now. She received her bachelor degree from Princeton, and then her PhD from Stanford. Her PhD work was actually done in fluid mechanics here in the mechanical engineering department. But after that, she actually joined the University of California, San Diego, and in the mechanical and aerospace engineering department. She started basically her career in the field that bridges mechanical engineering and fluid mechanics and medicine, specifically cardiovascular flow. She has been very awarded a number of recognition for her work over the years, including the NSF Career Award, the Burroughs Wellcome Fund Career Award, and she has been extremely prolific with her work and her publications, but also a real community leader by bridging sort of the development of tools and research in that area with spreading that with the community, with her codes and her algorithms that are actually available for everybody to use. I'll let Alison take over from here and introduce the session and the speakers.
Great, thank you, Gianluca. I'm excited to be here to introduce our first session in the Expo this year. And the first session is, of course, highly relevant to our current situation. It's focused on human-centered computational math related to health and disease. So, of course, never has this been more relevant than it is right now, as we're all affected by the outbreak of COVID-19. And ICME has been increasingly active in the space of computation related to health and healthcare in recent years. We're actively expanding our activities in healthcare, drug discovery, disease, and we're excited to have a number of new partners and initiatives in these endeavors. Looking globally at our activities, in terms of computational and mathematical methods, our activities span a wide range of various methods, from PDE simulations for biomechanics to AI for image processing and pattern recognition, stochastic simulations and uncertainty quantification, graph theory, natural language processing, and cloud computing for chemical biology. In addition, we can think of our activities in terms of spanning a wide range of biological scales, from organ scale, surgical planning and disease, to organization and dynamics of cell behavior, to molecular dynamics and genomics, and protein folding. And we're also, through these efforts, spanning a wide range of applications in terms of disease, from cardiovascular disease and cancer, all the way up to hospital logistics and operations, wearables, wireless devices and their associated data processing, protein-drug interactions, and of course, the spread of infectious disease, which is one of the topics we're going to talk about in this session. We're going to talk later a little bit about the AI for Good seminar series, and there's also a new initiative called the Human-Centered AI Initiative on campus, and these are both really efforts that are focused on the future of AI having a positive impact in society. So we'll go ahead and get started with this session. In this session, we're going to hear from 3 investigators who are working on really distinct and high impact areas in healthcare. The first talk is from my own lab. Gabriel Mayer is going to talk about deep learning to support patient-specific modeling in cardiovascular disease. And then from there, we're going to move to a talk from Julia Palacios, who's going to talk about analyzing molecular sequence data related to the current COVID-19 outbreak. And finally, Sylvia Palacios is going to talk about single-cell analysis for reconstructing the tumor microenvironment in cancer. So I'll go ahead and introduce the first speaker, Gabriel Mayer, who's soon to be Dr. Gabriel Mayer. He is a PhD student in ICME, a member of my own lab. He is graduating imminently, as in next month. And he was one of our first students who had the pleasure of doing his thesis defense over Zoom just a short while ago. He did his bachelor's and master's at TU Delft in the Netherlands, and then came to us to ICME for his PhD. He has accepted a position to work in AI at BlackRock following this. And his PhD work has focused on implementing deep learning methods for improved segmentation of medical image data in cardiovascular disease, to support our efforts in surgical planning and disease outcomes. And as you'll hear from him, he also made a significant contribution to the application of uncertainty quantification in our image segmentation process. So I'll turn it over to Gabriel. We're looking forward to your talk.
Great, thanks for the introduction, Alison. I'll share my screen now so everybody can see. Yeah, so as Alison mentioned, I've been working in her lab as a PhD student on applying deep learning methods to the cardiovascular modeling workflows that we use. And so that's what I'm going to be talking about today. So really a big motivation for our work comes from the fact that cardiovascular disease is really a very big global issue that impacts many people around the world, and then to a lot of people, I think the scale is not quite known. In fact, it's estimated that about 30% of global deaths is actually due to cardiovascular disease, right? So that's an enormous amount. And in fact, most of those are even due to heart attacks and strokes. So really just these two things are killing quite a few people every year. And in terms of monetary value, it's estimated this costs society around $400 to $500 billion a year. And so really this is quite an issue, and it's something that sort of should be addressed in sort of any way possible. And this is part of the reason that we're sort of focusing on these areas. And so recently a technology that sort of emerged as a possible avenue towards improving the way we can treat and study and analyze cardiovascular disease is actually being able to simulate the blood flow within a particular patient's blood vessels. All right, and so the way this works is that we use numerical processes and computational fluid dynamics to simulate the flow of the blood. And in particular, we use geometric digital models of the patient's blood vessels to be able to constrain the domain of the simulation so that we can get an accurate representation of what the blood flow looks inside a particular patient's blood vessels. Right, and so an idea of this is that this requires us to actually know what the patient's blood vessels look like, which of course you don't know this beforehand, right? And so what typically happens is that somebody will come in, they'll get either a CT scan or an MRI scan so that we can see the blood vessels. So that you can see a couple examples of those on the slide, and then we'll have to use a workflow to actually model these blood vessels, shown in the bottom left, starting with the image. We have to detect, where the blood vessel center lines are, what their shape is, and then from that we can create a 3D model and use that for a simulation. All right, and so this detailed workflow is pretty sophisticated. And so what Alison has done with her lab is developed an open source software called Simvascular, that exposes this process and makes it publicly available for everyone to use. So features like image visualization, image processing, model construction, meshing, all of that is, just implemented in this software and available. And in particular, we have about 4,000 users worldwide, with quite an active, development team working on this software. So, it's great to see that these technologies are spreading and that, people are taking an interest in these things. And so in particular for my research, one of the things about this workflow is that constructing the cardiovascular geometry for a particular patient can be quite time consuming and laborious, right? Finding these blood vessels in an image, detecting their shape, especially if it's not automated, has to be done by a person. And so that can take quite a while, especially, if you're modeling something with, say, hundreds of vessels. And so I've been looking into how can we use, machine learning and deep learning and other data-driven methods to automate this workflow. Especially over the years that Alison has been running her lab, they've built up quite a database of, example cardiovascular models and patient images. And so that's what I've been looking into, how to use these to actually automate this process. And I developed some convolutional neural networks that were able to, take this workflow and automate certain steps. So that's, shown in the first picture on the left. We have a neural network that, traverses these blood vessel center lines and is able to detect the local shape of the blood vessel to then produce a 3D model. And in the bottom left, we compared this network to the accuracy of humans and found that for most blood vessels, in fact, the neural network was just about as accurate as the agreement between experts. So showing that, indeed, these data-driven processes and, deep learning methods can very realistically model the performance of even human experts. So that was very interesting and promising. And in fact, we even went a step further. Using the neural networks, we were able to uncover sort of a new research direction that was previously sort of not possible. And that was actually to try and quantify, or to be able to sample, for a given image, what are all the possible blood vessels that could be in this image? Because these medical image volumes are, of course, pretty noisy. There's all sorts of scattering effects due to the, way these things are taken. Additionally, there can be some disagreement between human experts on what exactly the blood vessel is, differences in contrast absorption, all kinds of things, artifacts. And so really there isn't just one blood vessel for a particular image, but there could be many possible blood vessels. And so being able to, quantify that uncertainty is extremely important, especially, if you want to take these to a clinical provider and establish confidence in your simulation outputs, you need to know what those uncertainties are. And so it was very interesting that using these neural networks, we could actually, take a step towards being able to quantify this uncertainty. In particular, we figured out that adding dropout layers to the network would actually allow it to learn this uncertainty from the data. And then we could combine this with our path planning workflow to create a Monte Carlo approach for sampling full cardiovascular geometries, right? So this was very exciting.
1 minute.
And those are shown below. And so the results that came out, we looked at the flow profiles for some typical cases, and we saw that there was indeed quite some uncertainty in the flow outputs. So here we can see that, the pressure is affected, the wall shear stress, the velocity of the flow. And in particular, we were able to quantify sort of the effects over the geometry and see which areas were the most affected. And comparing to other sources of uncertainty, such as, for example, uncertainty in the input pressure, or the material properties, we saw that geometry uncertainty was indeed a very significant source, particularly for wall shear stress, where uncertainties could go up to, say, 20% in the smallest vessels. So that was, very interesting how deep learning enabled us to actually approach this new topic and showed us that the results were actually quite important and, relevant to the field. And of course, this was all made possible thanks to, many collaborators, and of course, my advisor, Alison, running her lab over the years and building up this data set. So a big shout out to all of them as well. Thank you.
Thank you, Gabriel. So I think we have time for a couple of questions. I can start out with one. Can you talk a little bit about how you might integrate your work on uncertainty quantification of the image data with other sources of uncertainty in efficient ways?
Right. Yeah. So that's a big thing, is that really there's, in any given simulation, there's not just one source of uncertainty. You have all these sources of uncertainty sort of going on at the same time. You have geometry uncertainty, but you also have input uncertainty, material properties uncertainty. And so, yeah, really what you would want to do is to be able to sample all of these at the same time. And so one thing you could do is you could take the Monte Carlo sampling procedure we have, and then on top of each of those model samples, you could overlay different sources of other uncertainties that you're interested in, right? So you could generate a set of model samples, and then for each of those models, run a couple scenarios with, say, a different pressure waveform. And this could show you, say, if you add pressure uncertainty on top of geometry uncertainty, how do those interact? Does it increase the uncertainty, or maybe there's like a modulating effect where it's not as bad? So that would be one way to sort of, combine those together.
Great. So Sylvia Plavridis is a professor of biomedical data science and radiology, and the chair of the relatively new Department of Biomedical Data Science here at Stanford. She leads a systems biology cancer research program that bridges genomics, biocomputation, imaging, and population sciences. And she's going to talk to us about reconstructing the tumor microenvironment interactions through integrative single cell analysis. Sylvia has, of course, won a number of awards. I think in the interest of time, we'll let her get started. But she has an impressive list of accomplishments, and I'm excited to hear her talk. Sylvia, go ahead.
Terrific. Well, I'm so delighted to be with you this morning. My short talk highlights some work from my lab in computationally reconstructing the tumor microenvironment. My lab is located in the Clark Center, which is pictured here, which I miss. I miss so much. I know we miss me on campus. We have two spaces, one for computational scientists and one for cancer biologists. So we truly are a hybrid lab. So you might be asking, what is a tumor microenvironment? When we think of cancer, we often focus on the malignant cells, but in actuality, the malignant cells cannot survive on their own. In fact, they're comprised, compared to many other cell types, they're actually very hard to culture from primary tumor samples. Malignant cells reside in a microenvironment comprised of many different cell types, many of which are not really fully characterized. And it's really in the interaction with these cell types that tumors develop, grow, resist treatment, and metastasize. My lab, among many others, is trying to understand the interactions in the tumor microenvironment. And our rationale is that if we can identify key interactions that can be therapeutically targeted, we will be more successful eradicating the tumor at its local site and even systemically. A recent example is immunotherapy, which enables the interactions between cytotoxic T cells and malignant cells. However, immunotherapy is not always effective, and we believe that's in part because the tumors are not just composed of cancer cells and immune cells, but they actually have other cells too. And understanding really the complex interaction between all the cell types is going to enable us to, I think, crack the nut actually on some of these immunotherapy approaches and really contribute to understanding disease progression and treatment response. So we're really enabled to tackle this problem because in recent years, there have been a number of technological advancements that enable single cell analysis. My lab is a heavy user of 3 platforms in particular. The first is CyTOF, where we study proteins at a single cell level using tools like CyTOF. In addition, we interrogate entire genomes as a single cell level using tools like the 10x Genomics platform to generate RNA-seq data. And most recently, we've invested in a multiplex immunofluorescence imaging platform developed at Stanford called CODEX to interrogate tissues with single cell resolution in situ, that is, without dissociating them into single cells, but actually studying them at the single cell level so that we can assess spatial neighborhoods and associate those spatial neighborhoods with specific tumor phenotypes. We apply a number of analytical approaches. We apply graphical networks to understand interactions where the nodes typically are the cells and they're connected together if they're signaling partners between the cells. We apply tree-based analysis to study cancer lineages. We know that cancer cells, among other cells, evolve under environmental conditions and they change their states. And so we look at that evolutionary process, identifying them through these lineages. We also apply dynamical models to study the evolution of the cancer cells in time as they move through these lineages. And finally, apply machine learning algorithms to identify features most associated with specific phenotypes, such as progression. So next, I'm going to show you 3 pretty complex slides, but I'll talk about them at a very high level, sort of focusing the work in our lab. Some very recent work is trying to spatially resolve cell-cell interactions in tumor microenvironment. Here in the first panel, we show something called a cell atlas. And this is a really exciting activity where we take single cells dissociated and we study them in terms of their global genome expression. and here we project them into a lower dimensional space. And each point here in the space is an individual cell from primary tumor samples, and we identify the cell types. And you can see there's at least 20 different cell types in this particular analysis. We couple this analysis with a proximal slice of the tumor where we look at the spatial neighborhoods in situ with the Codex imaging technology. In panel 2, we just highlight 4 different cell types just for simplicity's sake. And here we're beginning to actually deconstruct these spatial neighborhoods. In panel 3, we want to actually merge the single cell data with the spatial information because we want to eventually map all the high dimensional genomic data onto into the spatial context. In panel 4, we use the genomic data to infer interactions between cell types, and here we're illustrating the different, circles are really composed of nodes that are specific molecules that interact with different cell types. So the colors are actually different cell types, and there's interactions between specific molecular players between ligands and receptors. So what we see here, for example, is that the central, one central process in our study relates to a phenomenon called epithelial to mesenchymal transformation. And that is a process that's really very important in metastatic disease progression and even drug resistance. And so we've been spending a lot of time trying to deconstruct what is really happening in this process. So in the second vignette that I want to describe to you, it's a published work that appeared last year. We're really trying to understand this epithelial to mesenchymal transformation with a time series analysis. With this time series data, we developed these Markov models to understand trajectories. And then that's what's in panel 2 and 3. And in panel 4, we actually take primary specimens and we project them onto the inferences made through these time series data sets so that we can essentially maybe deliver a new clinical assay to phenotype clinical specimens in terms of this EMT plasticity. And then finally, we've been using the single cell data to understand how individual drugs perturb cell states. This is a pretty complex slide. It's a published work that you can look at, but the basic bottom line is that we have an algorithm which relies on these nested effects modeling to determine what drugs that have similar and differential effects in different single cells in a tumor in order to design an optimal combination of drugs for an individual patient. So panel 3 is where we applied this algorithm to a cohort of 30 pediatric patients to determine the optimal combination, taking into account really single cell resolution in terms of their drug response information with respect to intercellular signaling states. So that's a lot of information, I realize. I just want to acknowledge the people in my lab who do the work and many of my collaborators at Stanford, including our funding sources, specifically the National Cancer Institute. So thank you for your attention.
Thank you, Sylvia. That was fantastic. So it's my pleasure to introduce Julia. She is an assistant professor of statistics and biomedical data science. To give you a little bit of background, she did her PhD in statistics at the University of Washington and then did joint postdoctoral research at Harvard and at Brown. In her work, she seeks to address statistical questions, mostly in population genetics, epidemiology, and comparative genomics. And a lot of this involves probabilistic modeling of evolutionary forces and methods to handle big data. So she is going to talk to us about the phylogenomics of COVID-19, of course, an extremely relevant topic for today. Julia, please go ahead.
So this is very recent work. It's still not published, many of the results. But what we are basically doing is that we are exploring the available sequences. Can you go back? Yes, thank you. We're exploring the available sequences of people infected from the virus. So phylogenomics aims to combine model epidemiological and evolutionary processes together to understand the disease dynamics. And the goal in this talk is to understand patterns of transmission, so the spread of the disease around the world, estimate the mutation rate, and hopefully use also this information for surveillance. So at the moment, GSAID is this public repository that has all these sequences available. We have about 30,000 samples from around the world, and those are the sequences that we're going to be analyzing in this talk. Next. So at the moment, we have available data from around 80 countries, mainly from the UK and the USA. So the UK has about 20,000 samples available for analyzing. Next. And we also have this database is being updated every day. So at the moment, we have mainly sequences from March and April. So there is this lagging with the information because when the patient gets infected, then when he knows it and then he gets sequenced and the data preparation and then it's deposited in GSA, it takes at least a month for that to happen. So at the moment, we have many of the sequences in March and April that correlates with the number of cases around the world. So on the left side, you have this blue curve that it represents the number of cases in the world. Next. So you can use these sequences to obtain a rough estimation of the mutation rate, of the substitution rate. So in the x-axis, I'm showing you the sampling date of the sequences and how many days apart they are from the original sequence in China. And in the y-axis is the number of mutations these sequences have accumulated with respect to the original sequence in December from China. So when you do this and you plot all these sequences, all these differences with respect to the reference sequence, you can estimate the mutation rate by estimating the slope of this line that crosses the points. So if you do this very simple estimation, you can get a rough estimation of the mutation rate that for us is 2.29 mutations per month. Next. So how is that 2.29 mutations per month with respect to all the other living organisms? So in the y-axis here, we have the genome size and the y-axis the mutation rate. So these are all the living organisms. So humans are in the blue on the right side. And coronavirus is this bubble above RNA viruses. This bubble, it's within the bubble of other RNA viruses, so it's not outstanding. It mutates a little bit more. Next. But with respect to all the other coronaviruses, so the rate in terms of years, it corresponds to 2 × 10⁻³ per base per year. And this too is within the range of other coronaviruses. So it's within the coronavirus mutation rate. Next. So what we can do with all this analysis is to learn patterns of spread. This is a now popular figure from NextTrain that I'm borrowing. But basically, you can track the disease from the beginning. So at the beginning of the phylogenetic tree on the left side, all the sequences came from China, and there was the transmission only within China, but then it moved to Europe, and then it moved to the US in red on the right side. Next. So that is interesting, but we can also estimate the population size of the number of infected individuals, and that's what we're targeting here. So the nice thing about molecular sequence data is that the patterns of mutations inform us about population sizes. So to see that, what we do is that we compare sequences, we see how many mutations they differ. And that tells us more or less how far back in time these two sequences share a common ancestor. In this case, how recent these two infected individuals share a transmission, so that someone who infected these two individuals in the past. So of course, if there are many mutations apart, that's an indication that the sequences, the pair of sequences have a recent common ancestor very further back in time. And if they share a small number of mutations, this ancestor is very recent. And the second key aspect is that if we have a random sample from the population and these sequences all are very similar to each other, that means that the population size is not too large because the virus hasn't infected many individuals as to accumulate many mutations. But if, on the other hand, we observe that the common ancestors of the individuals in the sample are very distant, that means that the virus has accumulated many mutations, that means that it has infected many people. So this is how we infer the population sizes, so the number of affected individuals from sequence data. Next. So now, with respect to viral dynamics for the population is a structure, very simple model is to classify the individuals as susceptible, infected, and recovered. And the evolution happens in time. Every time someone gets infected or recovers or becomes again susceptible when the individual recovers. So what we can do is that we can imagine that we have our population evolving in time, and then we sample individuals from the infected pool, from the red circle. And then what we're going to do is want to trace back the ancestry of those infected individuals. Next. All right, so when we trace back the ancestry of the individuals, the rate at which two infected individuals share a common ancestor back in time is this function over here at the bottom of the slide, that it's proportional to 2 times beta of t, which is the transmission rate of time t, times the number of susceptibles divided by the number of infected individuals. At the beginning of the epidemic, it's fair to assume that the transmission rate is constant and the number of susceptibles is constant, in which case this rate is inversely proportional to the proportion to the number of infected individuals. So estimating these rates will tell us about the proportion of infected individuals in the population. Next. So this is the basic model. From sequence data, we want to estimate the population size by modeling this ancestry. Next. So we do this doing a Bayesian estimation. We approximate this posterior distribution of everything, all the parameters that we don't observe, and we do this through MCMC. Next. So one thing to consider is that there is population structure, and then that there's also correlation of the sampling with time. Next. And next. So I want to show you, these are the results from China. So the tree is on the left, and what we are recovering is on the right. This is our effective population size, which, as I said, is proportional to the number of infected individuals. And in the last plot, in the bottom right, the black line corresponds to the number of new cases. So as you can see, these estimations, the both that we have, correlate highly with the number of new infections. Next. So this is Australia. Again, it correlates very nicely. So we want to use this information. So at the moment, it's something proportional to the number of infected individuals. It's not the actual number of infected individuals. So we're working in coupling this with SIR dynamic models to get a better estimation of the number of infected individuals and compare this with reported cases. But for now, if you go next slide, France and Australia, Canada, next. All these different results that we are obtaining are very, very reflecting very well the publicly available data. Next, Germany. Next, the last one I think is New York in the USA. And next one is, thank you for going over quickly. Yeah, it's fine. So we are doing this for all the countries. California, we're doing very well. Next. And this is New York. So New York, we have a lot of information coming from state of New York, and it allows us to even like forecast what's going to be reported. So there's always some lagging in the reporting, the number of cases that is reported by the government. But from sequences data, we can even go a little bit further in the estimation. So as you can see, the trend is reflected later on in the publicly available data. All right, I'm finishing here. I'm running out of time. I'm happy to answer questions after my talk.
All right, thank you, Julia. That was fantastic and of course very timely. I think we're nearly out of time. We had one question come in from James Cross for Gabriel. If we can bring Gabriel back, just to describe your training data set.
Yeah.
And I think we'll have to leave it at that for the session because we're out of time.
Yeah, so the training data set we had was a data set of around 100 medical image volumes, so CT or MRI image volumes, along with the corresponding blood vessel center lines, their cross sections, and the 3D cardiovascular models that had been made by previous students in Alison's lab for doing CFD simulations. So that was the data set we had, and in particular, we used the center lines of the blood vessels and the image volumes to extract 2D images of the cross sections of the blood vessels at particular intervals, and that's how we were able to turn that into a more manageable data set for training with deep learning.
And Gabriel, maybe say how many 2D cross sections you ended up training on from those 100
Oh yeah,
patients.
yeah. So from the data set of about 100 image volumes, we ended up with about 16,000 images of blood vessel cross sections, and so that ended up being sort of sufficient to get the level of accuracy that we ended up finding.
Okay, great. We're going to leave the session there. I really want to thank all three speakers, Gabriel, Sylvia, and Julia, for a fantastic talks spanning various areas of healthcare. And I think we're off to a break for 10 minutes. Thank you, everybody.